Welcome again to today's webinar on how to visualize gene expression with morphological and spatial resolution using RNAscope Chromogenic assay. Before we get started, I'll give you a brief overview of the agenda for today's webinar. We'll start with an introduction to the RNAscope technology. We'll discuss the RNAscope assay workflow, how to get started with the assay, some of the best practices for performing the assay, and then I'll turn it over to Anushka for showing some publication-based applications of the RNAscope assay. We'll then go to discuss the BaseScope technology and some of the applications of the BaseScope assay. Again, that's going to be covered by Anushka. We'll take a quick look at how to analyze your data, and then we'll have FAQs that would be answered by the Q&A window, as Rhonda and I mentioned previously. Okay, so we'll get started. So RNAscope assay is an in situ hybridization assay, which is a specific and sensitive method that allows you to detect RNA biomarkers in cells and tissues with a morphological context. As you can see on the slide, it's a hybridization-based signal amplification system and has a unique probe design, which we'll get to in after a few slides. More importantly, it allows you to detect single RNA molecules, and it also allows you to detect multiple mRNA molecules at the same time. And then finally, the detection can be either fluorescent or chromogenic. For today's webinar, we'll be focusing on the chromogenic assays. And last but not the least, the assay is compatible with a variety of sample types, such as FFPE, fresh frozen, fixed frozen cultured cells, to name a few. So on the next slide, we'll look at the overview of the workflow. So the RNAscope workflow starts with a pre-treatment process, which is required to unmask the target RNA and permeabilize the cells. This is important to allow the next step to take place, which is binding of the target-specific double Z probes to the target RNA molecules. This would then be preceded by amplification, which would be sequential hybridization of amplifiers and labeled probes. You will then detect the signal from individual RNA molecules, which can be visualized under a microscope. And finally, you can count this dots or signal from RNA molecules to get gene expression. So the unique part of the RNAscope and the BaseScope assay is its double Z probe design. So as you can see on the slide, the double Z probe has a bottom region, which we call the lower region, which is going to be complementary to your target mRNA, and it would serve as a target-specific binding site, which is about 50 base pairs in length. This is separated from the upper region by a spacer. The upper region is about a 28 base sequence, 14 for each Z, which would serve as the pre-amplified binding site, and that is where your amplification tree will be built. A regular RNAscope probe or a target probe set would be a pool of such 20 ZZ pairs targeting your RNA region, which would be about 1,000 bases, or in other words, 20 pairs of such 50 bases for the double Z. Now, for any detection technology or any molecular detection technology, there are two aspects which are very critical. One is sensitivity, the other is specificity, and the RNAscope double Z probe design helps achieve both of those. So as you can see here, the process starts with binding of the double Z probe to your target sequence. This is then followed by binding of the preamplifier, followed by binding of amplifiers to the amplification tree, and this would then be preceded by detection using a labeled probe, which can be an enzyme or a fluorophore. So this amplification is what contributes to the sensitivity of the assay. The assay is also highly specific, and by that, what I mean is, in a scenario you had only a single Z binding to your target sequence, there will be no signal detected because the amplification tree will not be able to build in a case where you have only one Z binding to the target sequence. So this is what contributes to the specificity of the assay. So the two parts achieved here are sensitivity and the specificity of the assay. The other thing, as I mentioned on the introduction slide, is that you can look at multiple mRNA molecules at the same time. And this is what brings into play the probe channel nomenclature. So in order to look at multiple probes at the same time, the double Z probe has a tail sequence that is indicative of a channel designator. So as you can see here, there is on the left-handed side of the top part of the double Z C1 probe, which has a tail sequence. This tail sequence is different from a channel 2 probe, which is shown on the right here. And this is important because the tail sequence is what contributes to the specificity of the channel probe detection and ensuring there's no cross-contamination or cross-detection between channel probes when you're looking at multiple mRNA molecules at the same time. I would like to let you all know that for tail designation, the RNAscope and the BaseScope assays use C as a tail designation or channel designation, whereas the high-plex assay, which will not be unfortunately covering for the webinar today, uses T for tail designation. We have, up till today, more than 2,700 publications since 2011, which is when the first publication using the technology was published. We had nearly 800 peer-reviewed publications in 2019, and all this metrics essentially shows the wide acceptance of the technology in the scientific community. If we were to break it down by the research area or research focus area, so to speak, here's what the graph would look like, with cancer and neuroscience being the two top areas where our technology has been used the most, but that doesn't take away the application of our technology for other fields such as for infectious diseases, developmental biology, immunotherapy, to name a few. Another interesting fact about the publication base we have is over a quarter of the RNAscope papers are published in some form of the big three journals, so that is from Nature, Cell, or Science. On the next slide, we'll review all the different BaseScope and RNAscope assays that ACD offers. To break this slide down, we'll split it into the first two columns to start with. So the first two columns show the chromogenic RNAscope assays that are available. So you can have chromogenic red or brown single-plex detection, which would be using either DAB, HRP for brown, and phosphored ALP for red. And you also have the option of looking at two mRNA markers simultaneously using the chromogenic assay, which would be RNAscope Duplex assay, which would detect two markers as red and green. And finally, both these options can be done manually, and they're also compatible on the LikeABond RX and Ventana Discovery Ultra platforms. We'll next look at column number 3, 4, and 5, which is about the fluorescent assays. So we have fluorescent multiplex assay, fluorescent multiplex version 2 assay, and the high-plex assay. The difference between the three is fluorescent multiplex allows you to do 3 targets. The version 2 of the assay allows you to do 4, which would require use of opal dyes. And then the high-plex assay, which is our newest product on the market, allows you to do 12 RNA targets in a single tissue section simultaneously. And then finally, on the far right, the last two columns, we have the BaseScope assay. Again, we have the two versions for the chromogenic BaseScope assay. You can visualize it as a single plex assay using phosphored, and you can also do a duplex quantification or detection using phosphored and green to look at two mRNA targets simultaneously. At this point in time, only the single-plex BaseScope assay is compatible with the automated platforms like LikeABond and Ventana Discovery. As I mentioned on the title slide, the focus for today's webinar will be on chromogenic assays. So we'll focus on RNAscope and BaseScope chromogenic assays. We'll next look at the amplification technology for the RNAscope assay. Shown on this slide is the amplification technology for a single-plex RNAscope assay, red or brown. On the left is shown the amplification setup for the red assay, where you have ALP or alkaline phosphatase enzyme being used, and phosphored would serve as a substrate to detect the signal as a red chromogen. Please note the phosphate chromogen is sensitive to alcohol and is not recommended for tissues with high alkaline phosphatase activity. On the right is shown the amplification for the single-plex brown assay. In this case, the enzyme is going to be HRP, and DAB would serve as a substrate to detect the signal as a brown chromogen. The brown assay is not recommended for pigmented tissues, so please take a note of that. On this slide, we are looking at the amplification setup for the duplex assay. In this case, you will obviously have red, as you had for the previous slide, detected using ALP and phosphate, and the green chromogen will be detected using the HRP enzyme. Very important point to note that in this scenario, or with the duplex assay, channel 1 probe will be detected as a green chromogen or a green dot, and channel 2 probe will be detected as red chromogen or red dot. And finally, please take a note that the green chromogen is both alcohol and water sensitive. We'll now dive into some of the tips on how to get started with the RNAscope assay. So before you get started, here are some of the important recommendations that we have. We recommend that you review the checklist, so ensure that you have all the recommended materials, have the user manual, also have the technical notes for sample preparation if applicable. And more importantly, we would like to emphasize on the fact that we would urge you to contact technical support for specific tips and tricks to get started using the email address shown at the bottom of the slide. Here are some of the recommended materials that we would recommend that you have before you get started. Just to highlight a couple of them on the slide, we recommend using SuperCross Plus slides because these are chart slides and help prevent tissue detachment during the assay. Use the high-DZ oven and accessory because the hybridization oven from ACD not only helps maintain optimal temperature, but also helps maintain optimal humidity, and both these parameters are critical to the success of the RNAscope assay. Here are snapshots of the reagent kits that you would require from ACD. Shown here are the pre-treatment reagent kits, the detection kits, the wash buffer for washing steps, and then control and target probes, which would be in channel 1 and channel 2. Please note that the wash buffer is provided as 50S, and if you notice precipitation, we recommend warming it up for 20 minutes at 40 degrees Celsius. We also recommend that you warm the probes at 40 degrees Celsius for 10 to 20 minutes before use. And finally, please allow the kits to acclimate to room temperature before running the assay. Here are some of the recommended accessories. Shown here is the hybridization oven. On the right and on the left, you see the easy batch tray for slide processing. When using the oven for the hybridization steps, please ensure that you have the oven turned on for at least 30 minutes at 40 C so that it can acclimatize to that temperature. And then make sure that you have the humidifying paper wet during the assay in the hybridization tray to make sure you maintain optimal humidity. We'll next look at some of the guidelines on how to prepare your samples. Shown on this slide are sample preparation guidelines for FFPE, fixed frozen, and fresh frozen samples. For FFPE samples, we recommend fixing your tissues for 16 to 32 hours at room temperature and 10% buffer formalin. You can store these sections at room temperature up to 3 months and at 4C for long-term storage. For FFPE, we recommend sample thickness of 4 to 6 microns. For fixed frozen samples, we recommend fixing it in either 4% PFA or 10% formalin for 24 hours at 4 degrees Celsius, and these can be stored in -80 freezer for up to 3 months in an airtight container. And the section thickness recommended for fixed frozen is 7 to 15 microns. For fresh frozen samples, we recommend freezing the tissues within 5 minutes of harvest using either liquid nitrogen or dry ice. And again, this can be stored for 3 months in an airtight container if they are put in a -80 freezer. And the sample thickness we recommend for fresh frozen is 10 to 20 microns. For additional sample types, if you're working with, say, cultured cells, cold mount, or other sample types, again, please contact technical support for further assistance. Shown on this slide is the pre-treatment recommendation for different sample types. For FFPE samples, the standard pre-treatment would include 15 minutes of target retrieval or antigen retrieval, and the protease used would be Protease Plus. For fixed frozen samples, we would recommend doing target retrieval for 5 minutes, and they would get Protease free, which is a stronger protease than Protease Plus. For fresh frozen samples, since these are not draw fixed, would not require target retrieval to reverse the formaldehyde cross-links, but they would get Protease 4, which is our strongest protease for pretreatment. And finally, for cultured cells, again, since these are not draw fixed, no target retrieval or antigen retrieval is required, but you would use Protease 3 with an appropriate dilution to figure out which dilution works the best for your particular cell type. We'll now look at how we can perform the assay with regards to the workflow on the timelines. So the RNAscope assay workflow can be done using 3 different options shown here. The first option is obviously you can do the assay in a single day where you prepare your samples, pre-treat your samples, proceed with probe hybridization, and then do the detection. The assay can also be split into 2 days, and there are 2 options for that. Option 1 would be that you stop after pre-treatment on day 1 and continue with probe hybridization and detection on day 2. Or you can use option 2, where you would prepare the samples, pre-treat, finish probe hybridization all on day 1, and on day 2 you will perform the detection part. We'll now look at the assay workflow. Shown on this slide is the red assay workflow for FFPE samples. As you can see, it's taken up into 3 major parts: pre-treatment, hybridization, and amplification, and counterstrain and mount. So the assay will start with pre-treatment, which would include baking, deparaffinization, hydrogen peroxide step, target retrieval to reverse the formaldehyde crosslinks, Protease Plus to the extracellular matrix, which would allow the probes to gain access. So then the next part would be hybridizing with your probes for 2 hours at 40 C. This would be followed by a series of amplification steps. And then finally, you would proceed with red color detection or red chromogen detection. And once the detection is done, you will proceed with counterstaining using chematoxylin, followed by bluing, air dry, and mounting. The total assay runtime for the singleplex red assay for FFPE samples is around 9 hours. Also, please note an optional stopping point shown here, which is after the target probe hybridization, you can stop and store your slide in 5x SSC overnight at room temperature. Shown on this slide is RNAscope Duplex assay workflow for FFPE sample. Again, the pretreatment part remains the same as for the singleplex assay. After the pretreatment, you hybridize with the Duplex probes, and then you would do a series of amplification steps, followed by the red color detection, and another series of amplification steps, followed by the green color or the green chromogen detection to detect the second mRNA. And then finally, you will counterstain and mount the slides. The total assay run time for the Duplex assay is roughly 11 hours. We'll next look at some of the best practices for the RNAscope assay. We recommend that you start practicing your technique using the control slides first. So this is what we call a technique check. In this scenario, what you will do is you will use our control slides, HeLa for human origin or 3D3 for mouse origin, and stain the slides with ACD provided positive and negative control probes. Once this is done, what you should expect is you should expect to very little background with the DAPI negative control probe and robust staining with the positive control probe, which is for the PPIB gene shown on the bottom image here. If this works out well, then you should go ahead and run your tissue samples with control probes. If not, we recommend that you verify the assay technique. So once the technique check has been done, as I said, the next step is to qualify your sample or do a sample RNA quality check. So again, in this scenario, you would run the positive and the negative control probes on your target tissue samples. And again, you should expect no to very little background with the negative control probe staining and robust staining with the positive control probe, which would qualify the RNA quality for the sample. If this passes, then we would recommend that you proceed with the target probe. If not, you should need to perform assay optimization or acquire new samples to check the RNA quality. Again, we would recommend that you start with recommended conditions, and for any additional assistance, please contact technical support at support.acd@bio-technique.com. I'm now going to turn this over to my colleague Anushka, who will go over some of the applications of the RNAscope assay.
Thank you, Tapan. As Tapan mentioned, today we'll be focusing on the RNAscope chromogenic assays and the BaseScope chromogenic assays. In this slide that you can see on the screen, we have detailed the different applications and research areas in which the RNAscope technology is applicable. The RNAscope technology encompasses both the RNAscope assays and the BaseScope assays. In the central panel, you can see the different target types that can be analyzed using these assays, which include non-coding RNAs, splice variants, GPCRs, point mutations. It allows you to visualize RNA and protein simultaneously using the dualish IHC workflow. This technology also allows you to validate single-cell RNA sequencing data and spatially map your gene expression. Because of the wide range of targets that can be detected using this technology, it has been widely used in different research areas. As you can see, it has been used in basic science research, for neuroscience applications, in stem cells, developmental biology. It has also been used for infectious disease research, especially for emerging pathogen research, where developing antibodies can take several months, but designing an RNAscope probe against an emerging pathogen is pretty rapid and allows quick research. In addition, we also support cancer and clinical research and biopharma discovery and development. In the biopharma discovery research, we've been widely used in preclinical models to assess the safety, efficacy, and toxicity of upcoming cell and gene therapies. Next. Now let's look at a few RNAscope chromogenic assay applications. We'll start by looking at one of our first applications for RNAscope using the chromogenic red detection kit. In this particular example, this group of researchers wanted to study the transcription factor ARNTL2 in lung adenocarcinomas. Now, this transcription factor is known to promote the expression of certain secreted factors that are significant in promoting malignancy and metastasis. Now, looking at the expression of this particular target, you can see in the central panel that this was significantly upregulated in poorly differentiated lung adenocarcinomas compared to well-differentiated tumors. And this really highlights the single molecule and single cell resolution that is offered by our assays. It further shows that the expression observed using RNAscope was correlating to what they observed by using the microarray analysis quantification. Next. Here we are looking at an example of RNAscope being used for infectious disease research. In the current environment, it's become very important to use the right tools to quickly get results on emerging pathogens. And this is a great example wherein in this particular publication that was published in Science, used RNAscope to study the efficacy of a vaccine they developed against MERS-CoV virus. In this particular example, they have used the Canon model and they developed a vaccine against the MERS-CoV and studied the efficacy of the vaccine, as you can see, by looking at the expression of the MERS-CoV virus using RNAscope probe. In the top panel, they have a vaccinated animal and a control animal. After giving the vaccine, these animals were exposed to the virus. And as you can see, the animals that were vaccinated show a very low expression of the viral RNA, but the control vaccinated or not vaccinated animals show very high expression of the viral RNA, indicated an active, strong infection by MERS. So this kind of showed them that the vaccine that they had developed was pretty successful in imparting immunity. In the bottom panel, they have compared the expression or the detection of MERS-CoV antigen using IHC to MERS-CoV RNA using ISH. And if you look at the signal, it shows you that although the patterns correlate, the expression observed by the ISH assay is much stronger, highlighting the sensitivity of the RNAscope assay in detecting viral RNA. Next. This is another example highlighting the use of the RNAscope Brown assay in infectious disease research. These group of researchers wanted to study the pathogenesis of Zika virus, specifically in the eye tissue. And for this particular experiment, they injected a mouse model with active Zika virus and then isolated the eye tissue to look at the expression of the viral RNA within the eye. As you can see in the central panel, compared to the mock infected, the Zika infected eye showed presence of the viral RNA in the different layers of the retina, as well as in the cornea. Expression was also observed in the optic nerve tissue. Now, if you compare it to the mock infected, which is completely negative for any kind of Zika infection, it shows you the specificity of the RNAscope probes in identifying your target viral pathogen. Next. Now, this is a very interesting example because it shows the application of the RNAscope Brown assay in cell therapy. This group of researchers from Innate Pharma were developing a NK cell therapy called NK cell engagers to increase the anti-tumor immune response within tumors. So they developed a bispecific and trispecific antibody to promote the infiltration of NK cells into antigen-positive tumor cells. And they used the RNAscope assay to detect NCR1, which is a marker of NK cells. If you look at the central RNAscope panel, you can see a animal xenograft tissue that was injected with the control treatment. And on the right-hand side, you can see a panel which had the treatment injected. And if you compare the expression of NK cells, you can clearly see that the panel on the right-hand side, which had the NK cell engagers injected, shows a significantly higher degree of NK cell infiltration within the tumor. And this shows you that the NK cell engagers were indeed successful in mounting a stronger immune response against the tumor antigens by increasing the infiltration of NK cells within the tumors. If you focus on the bottom right-hand side panel, where you see the graph of tumor growth with the different treatments, you can see that they used the bispecific antibody, the growth of the tumors was significantly retarded compared to that of the control treatment. So this kind of helps them understand that they are bispecific and trispecific NK cell engagers were in fact successful in recruiting more NK cells to the tumors. Next, please. In this particular example, we're now going to look at how the RNAscope Duplex assay was used to study the cellular states within a glioblastoma tumor. Now, this particular group from Harvard and MIT wanted to understand the different cellular states of a glioblastoma tumor. By doing single-cell RNA sequencing, they identified that there are 4 different cellular states in which a glioblastoma tumor cell can exist. They associated different markers with these individual cellular states using the RNA-seq technology. But they wanted to visualize these cellular states in vivo, and they utilized the RNAscope Duplex assay to visualize these different cellular states within the glioblastoma tumor. But what they observed was some of these markers were co-localizing in some cells, indicating that some cells not only exist as a single cellular state, but can exist in a hybrid state and show expression patterns belonging to more than one cellular state. And this was a very interesting find because using RNAscope, they were actually able to validate that these hybrid cells exist by showing the co-localization of these markers that you see in the right-hand side panel. So this was a great example of how RNAscope was used as a validation tool for single-cell RNA sequencing. Next.
All right, thank you, Anushka. So those are some of the really cool applications of the RNAscope assay, especially shown in publications. We'll now take a look at the BaseScope assays. Before we begin, let's look at some of the differences between the RNAscope and the BaseScope assays. So as I discussed, RNAscope assay, the probes are about 20 ZZ pairs in length, but the BaseScope assay is designed to detect shorter RNA targets, and therefore the probes are about 1 to 3 double Z pairs per target. Both of these technologies allow single molecule detection. RNAscope is recommended for longer transcript detection to transcripts that are 300 nucleotides or longer, whereas BaseScope is recommended for detection of transcripts or sequences such as exon junction, highly homologous short sequences, which would be about 50 to 300 nucleotide base pairs in length. And then both of these assays can be done on the automated or the manual platform. However, at this point in time, the RNAscope assay can do up to 4-plex, whereas the BaseScope can only do single-plex to dual, and only the single-plex assay is compatible on the automated platform. Just to give you an overview, the workflow remains very identical to the RNAscope assay, where you do the pretreatment, followed by binding of the target-specific double Z probes, followed by amplification using sequential hybridization of amplifiers, visualization of the RNA molecules under the scope, and quantification. There are 3 points that are different from the RNAscope assay. One is RNAscope assay would require a weaker protease, or in other words, the BaseScope assay would use a stronger protease for detection. This is because we're now trying to look at shorter transcripts. Again, as we're looking at shorter transcripts, the probe length is going to be shorter, wherein it's going to be 1 to 3 ZZs in length. And then because we're looking at shorter transcripts, there are additional amplification steps for the BaseScope assay, which is required in order to detect shorter RNA transcripts. The amplification technology for the single-plex red assay is still very similar to the RNAscope assay, where the detection for the single-plex red assay for BaseScope would be just like RNAscope, using ALP as the enzyme and phosphate as the substrate. Again, I would like to reiterate that the phosphate chromogen is alcohol sensitive. Here is an example of the BaseScope red assay workflow for FFPE samples. Again, as was for RNAscope assay, the process starts with pretreatment, followed by hybridization and amplification. Now, as you can note here, compared to RNAscope assay, where you had only 6 amplification steps, the BaseScope red assay has 8 amplification steps, and this is just to reiterate that this is the additional amplification boost which is required to detect shorter RNA transcripts. After the amplification steps are done, you would do the red color detection, followed by counter stain and bluing, and then you mount the slides. The total assay runtime can be 9 hours or longer. The BaseScope Duplex assay, which is red-green detection, again is very similar in principle to the RNAscope assay, where you would have ALPV-based detection for the phosphorid chromogen and SRP-based detection for the green chromogen. Again, I would like to reiterate the fact that with the Duplex assay, the green chromogen is detected for the C1 or the channel 1 probe, and the red chromogen is detected for the C2 or the channel 2 probe. And the green chromogen is both alcohol and water sensitive. Again, shown here is the Duplex assay workflow for FFPE samples. Again, the workflow starts with the pre-treatment, followed by hybridization of the target-specific probe, followed by a series of amplification steps. You would then do the red chromogen detection, followed by another series of amplification steps, and then you would do the green chromogen detection for the second mRNA, counter stain in mouse. The total assay runtime is 11 hours or longer. Again, especially for the BaseScope assays, since these are longer compared to the RNAscope assays, we would recommend that you utilize the optional stopping point, which is after the target probe hybridization, you can stop and store your slides in 5x SSC or sodium saline citrate buffer at room temperature overnight, come back the next day and start with the amplification steps. We'll now turn it back to Anushka to look at some of the chromogenic assay applications for the BaseScope assay.
Thank you, Tapan. As Tapan mentioned, the BaseScope assay is a specialized assay that can be used for detection of short targets, splice variants, and point mutations. I'll be showing examples of each of these. So let's start with how RNAscope has been used for visualizing short target sequences. Now, for the first example, something that has become really important in the last decade is the development of engineered T-cell therapies. And for this particular therapy, it's important to identify a specific clonal population of T-cells. And for this, you need to have a specific probe designed against the CDR3 sequences of the T-cell receptors. And using the BaseScope assay, we can design a specific CDR3 probe for your PCR of interest. Next. In this particular example, this is a paper that was published last year by the Bauko lab showing the use of BaseScope assay in detecting a specific population of T cells. In this example, a patient was being treated for melanoma using the anti-PD-1 therapy, and the patient responded really well to the therapy until they developed some adverse events related to the anti-PD-1 therapy, and this induced encephalitis. And ultimately, this encephalitis was fatal for the patient. So these researchers wanted to really analyze what population of immune cells induced this encephalitis due to anti-PD-1 therapy. And for that reason, they did PCR sequencing on this encephalitis tissue and found that there was a specific sequence that was most prominently expressed in this tissue. They developed a BaseScope probe against this sequence and visualized this PCR in that tissue. And what they found was that when they co-localized this specific PCR sequence with either CD4 antibody or CD8 antibody, they saw that the CD4 and TCR co-localized, but the CD8 and TCR sequence did not co-localize. This told them that the population that was probably inducing this encephalitis was a CD4 positive T cell population. On further analysis, they were indeed able to confirm that this encephalitis was predominantly induced by a CD4 positive memory T cell population. So using the BaseScope assay, they were able to validate and visualize this population of T cells that was causing the adverse events induced by the anti-PD-1 therapy. Next. Now we are going to look at an example of how BaseScope Duplex assay was used to visualize CRISPR-mediated gene deletion. Using a probe design strategy, we were able to design a probe specifically for the wild type transcript of a particular gene and then a specific probe to the edited version of that transcript. And using two different probes and the BaseScope Duplex assay, we were able to visualize both the wild type and the edited version of the transcript in our target tissue. Moving on to the next slide, you can actually see that when we looked at the expression of both these wild type and edited transcripts in our target liver tissue, on the left-hand side panel in the non-edited liver tissue, we could only detect the wild type transcript indicated in green, and we could not detect any edited transcript as you would expect. On the right-hand side panel, on the other hand, which was the tissue that had the edited transcript, we can see majority expression of the edited transcript and some degree expression of the wild type transcript. Again, highlighting that our BaseScope probes are very specific against these short targets, which can be very challenging to detect by using some other technologies. So using this BaseScope assay, we were able to visualize both the wild type and the CRISPR edited gene in our target tissue. Next. Now we move on to an example of how BaseScope can be used to visualize splice variants of important target genes. HER2, as we all know, is a significant target in breast cancer. This group of researchers wanted to stratify patients based on the presence of the delta 16 HER2 splice variant. Since individuals with the delta 16 HER2 splice variant respond really well to Herceptin therapy. Now, they used a probe design strategy wherein they designed a probe against the exon 15 and exon 17 junction so that they could specifically detect tissues that had the delta 16 HER2 splice variant. On the other hand, they also designed a probe that spanned the exon 15 and exon 16 junction to detect the wild type HER2 gene. And third, they designed another third probe that spanned across exon 17 and exon 18 to detect both the wild type and the delta 16 splice variant. So, Tapan, if you click again, here when they used all the three probes, on the far right-hand side panel, you can see that they were able to identify samples that have the delta 16 splice variant. In the middle, using the wild type probe, they were able to identify the wild type HER2, and the common region probe was able to detect both. But if you compare that to the IHC staining, it was not able to distinguish between the splice variant and the wild type target. Again, highlighting the significance of using a BaseScope assay to identify specific splice variants for patient stratification. Next. Now, here is a very interesting example, again, of detecting splice variants using the BaseScope assay. Now, we have moved on to a neuroscience application where DL gap circular RNAs are predominantly expressed in the developing brain. Circular RNAs are formed due to back splicing, and here we have designed a specific probe against the exon 9 and exon 7 junction, which is found in these circular DL gap RNAs. And so we wanted to specifically detect the circular RNA and not the linear form of the DL gap RNA. In this example, we've used the BaseScope assay in combination with IHC for MAP2 to detect the expression of DL gap in the dendrites. As you can see, the red punctate dots in the zoomed in image, can you go back? The red punctate dots in the zoomed in image in the bottom right corner indicate the single cell and single molecule resolution offered by RNAscope. Now, an interesting property of our red chromosomes is that it can fluoresce under the microscope. And this particular image, after staining using our BaseScope assay, we counter-stained with DAPI and visualized the slide under the fluorescent microscope. And you can see that the DL gap staining is now visualized as fluorescence staining under a fluorescent microscope. And this can be very significant for targets that are expressed at a low level, if you want to get a much better contrast for your staining. Next. Our final application for the BaseScope assay is using BaseScope for detection and visualization of point mutations. On the screen are listed the number of publications we have using our BaseScope point mutation probes, and we have over 15 internally validated point mutation BaseScope probes for some of the most commonly found point mutations for diseases. So if you're really interested in studying any of these point mutations, do reach out to us and we'll be happy to provide you with the validated list of point mutation BaseScope probes. Next. We will now look at an example of how using a BRAF V600E specific BaseScope probe, we were able to identify this particular mutation in a cell line in-house. So we have used two different cell lines, the SKML28 melanoma cell line, which has the V600E BRAF mutation, and then the CHLA1 cell line, which has the wild type BRAF. So when we use the mutant BRAF probe and compare it to the wild type BRAF probe, you can see that positive signal is only observed in the SKML cell line, which has the point mutation. And there is no signal observed in the BRAF wild type cell line. Again, highlighting the specificity of our BaseScope probes in detecting point mutations. And as I mentioned, we have validated several different probes using our technology. So if you're interested in a specific point mutation, do reach out to us. Next. Finally, I want to highlight a very interesting application of the BaseScope assay, and that is for detection of pre-microRNA. MicroRNAs, as you know, can be important regulatory RNAs that can be significant in a number of pathologies, including cancer. In this particular example, this group wanted to study the expression of MIR135B in gastric cancer. Inflammation, chronic inflammation can be an important factor in inducing malignant transformation of gastric epithelium cells. And the role of microRNAs in this malignant transformation is not that well understood. So they wanted to study the expression of microRNAs and understand if they played a role in inducing this malignant transformation. And what they observed was in tissues that had gastritis, that is inflammation of the gastric tissue, or in gastric cancer tissues, they found increased expression of MIR135B, and they found a parallel increase in Ki67 expression, indicating that these tissues that had higher expression of MIR135B are also highly proliferated. So the change in gene signature that you get after malignant transformation can be regulated by a microarray such as the MIR135B. So using the BaseScope assay, they were able to identify the pre-MIR135B in this gastritis tissues and gastric cancer tissues. And with that, I hand the platform back over to Tapan.
Thank you, Anushka. So this takes away nicely into a new product that's coming soon, which is going to be RNAscope. Shown on this slide is staining for mature microRNA 124.3p in the hippocampus, cerebral cortex, and cerebellum of the brain, and then liver and kidney. So far, we've seen that the RNAscope demonstrates high specificity and sensitivity in brain, liver, and kidney tissues, as you can see here. And the RNAscope can be used for detecting mature microRNAs, ASOs, and siRNA. So this product is coming up soon, so please stay tuned. With that, we're going to get into working with some of the non-standard sample types, whole mount tissues, free-floating, bone, organoids, or skin, to name a few. I would like to emphasize on the fact that if you're working with this non-standard sample types, we urge you to contact technical support via the email address shown at the bottom of the slide here for suggestions and guidance before you get started. But in the next few slides, Anushka is going to show you applications for working with RNAscope on this non-standard sample types to show you that indeed the RNAscope assay is compatible with this non-standard sample types as well.
Thank you, Tapan. So as a first example, we are going to look at how RNAscope is used for detecting gene expression in the bone tissue. So here the researchers wanted to understand the role of SHP2 in differentiation into osteoblasts. So they looked at the expression of several targets that are regulated by SHP2 using our brown chromogenic RNAscope assay. And they saw that when SHP2 was knocked out of the tissue, there was downregulation of some of these targets that are important in osteoblast development. Next. Here's an example of RNAscope being used in the skin tissue. We have seen a lot of publications recently of RNAscope being applied in the skin sample, but as you all know, it's difficult sample to work with because of the processing that it requires. In this particular example, these researchers are studying the psoriasis model and trying to identify expression of cytokines that are upregulated in psoriatic skin. Steroid is a very common therapy that has been used to treat psoriasis, but it can have some debilitating effects. These type of researchers wanted to study the effects of vitamin D analog like calcipotriol in decreasing some of the symptoms of psoriasis. Now, the role of vitamin D3 has been well established, but the mechanism underlying it is still not very well understood. So using RNAscope red assay, these researchers studied the IL-23, IL-17, IL-36 signaling pathway to see the expression of IL-36 cytokine. And as you see in this inducible model on the right-hand side panel, when psoriasis was induced, the expression of IL-36 alpha and gamma is significantly upregulated. And in the bottom panel, when this animal is treated with calcipotriol, the expression of IL-36 is significantly reduced, showing that it is in fact reducing inflammation and hopefully subsiding the effects of psoriasis. Next slide. In the same study, they wanted to compare the effects of calcipotriol to that of dexamethasone, which is a steroid. And if you see in the panel, they treated the psoriasis individually or independently with calcipotriol or with dexamethasone or with a combination of calcipotriol and dexamethasone. And in the second column or the second panel, you can see after induction, expression of IL-36 goes up. After calcipotriol treatment, it goes down, similar with dexamethasone, but using a combination of both seems to have a cumulative effect on reducing the expression of IL-36. So using the RNAscope assay, we were actually able to visualize the expression of IL-36 in different regions of the epidermis. And an important thing to emphasize here is that this technology allows you to visualize the cellular source of these secreted factors, such as cytokines and chemokines within different tissues. Next. Finally, I'm going to talk about the use of RNAscope in studying gene expression in organoids. In this example, this group of researchers developed an organoid library for pancreatic cancer tumors, specifically for pancreatic ductal adenocarcinomas. They wanted to see the role of Wnt signaling pathway in pancreatic cancer progression. So for that purpose, they developed an organoid library and studied the expression of different Wnt ligands using the RNAscope red assay. And as you can see in the panel, there is a very high heterogeneity of Wnt expression within different tumors. This highlighting that the role of Wnt in pancreatic cancer progression can be different for different tumors. And using RNAscope assay, they were specifically, again, able to identify which cell types within these organoid tumors are actually producing these Wnt ligands. So with that, I transfer the platform back to Tapan.
Thank you, Anushka. So one of the other really interesting applications of the RNAscope or the BaseScope assay, as Anushka showed on some of her slides, is the capacity to perform dual RNAscope-ish IHC. Shown here is the workflow for the dual RNAscope-ish IHC combination. We recommend that you perform RNAscope first always, and then go to the IHC part or IF part. Some of the tips for this dual-ish IHC combination would be it's only compatible with either the RNAscope 2.5 HD Red or BaseScope V2 Red assay. It's also compatible with the automated assays for the Red assay, but it's not compatible with the Chromogenic Brown or the Duplex assay. All of these protocols would require optimization, and as I said, perform ISH first. We also recommend that you optimize ISH separately before combining it with RNAscope assay. And finally, when you're choosing your protein target for immunostaining, we would recommend using highly expressed protein to do protein degradation during the ISH protocol, and preferably using antibodies that are validated. Shown on the next slide is a great example of dual RNAscope-ish IHC staining with the chromogenic assay. Shown on this slide is PD-L1 RNA staining using ISH in red, and for CD5 protein with IHC in green or teal here. Again, showing a great example of dual-ish IHC application. We'll now look at some of the guidelines for quantifying your RNAscope and BaseScope data. Some of the key questions with regards to the signal is what is a typical signal? What does a dot mean? What is the significance of the dot size? And what's the difference between a dot and a cluster? Shown on this slide is the difference between dots versus clusters. As I mentioned at the start of my webinar, mRNA molecules with the RNAscope or the BaseScope assay are going to be detected as punctate dots, which is shown by yellow and blue arrows here. You can have clusters which would result from signals that come from mRNA molecules that are in close proximity, shown by the brown arrow. And then finally, I would like to point out that you can have variation in either dot size or intensity, which is related to the differences in the number of ZZ probes that are bound to the target RNA. So as you can see here, the blue arrow indicating a dot is way lighter than the dot that is indicated by a yellow arrow here, which is potentially due to the number of ZZ probes that are bound to the target. However, an important fact to remember is each of these dots represent a single mRNA transcript, and at the end of the day, it's the number of the transcripts that is important and not the intensity or the size of the dot. Shown on this slide is how you can perform data analysis using the semi-quantitative method. The semi-quantitative method essentially has a table shown here, which would basically isolate the staining into different scores. So starting from a score of 0, which would mean no staining or less than 1 dot per 10 cells, and then incrementally going towards a score of 4, which would be more than 15 dots per cell and more than 10% dots in clusters. Shown in the bottom here is a representation of a staining for a score of 0 to 1, 2, 3, and finally the score of 4. You can also perform data analysis using the quantitative method. This can be done using open source image analysis software such as ImageJ from PG or Cell Profiler. Here are the links to download the software. And then here's what you can do with the quantified data. You can present them as total cell count, probe.count, number of positive cells, to name a few. In order to do the data analysis, we have multiple resources available for data analysis. We have guidelines on how you can perform the RNAscope assay quantification, and we also have technical notes on how to perform quantitative data analysis for your RNAscope and BaseScope data using either ImageJ or Cell Profiler. Finally, this is the last few slides. with the current scenario, the demand for tools to perform COVID-19 research has been high, and we are offering tools to researchers who are doing COVID-19 related research. Shown here is kind of a snapshot of what you can do with our COVID-19 offering. I mean, obviously, you can do the viral detection with the COVID-19 probe, but you can also use a wide array of other probes to detect other targets which are associated with the pathogenesis of the COVID-19 probe. So you can use probes that are for receptor and protease expression, and then you can also use probes for the immune response to look at the cytokines. And then finally, you can visualize this with the power of the RNAscope assay, where you can look at this in physiological and pathological tissues, but you can have the morphological context with specificity and single molecule sensitivity. So if you're interested in learning more about our offerings for COVID-19 research, please feel free to reach out to us. And obviously, with the current scenario where we're living in very challenging times, if you happen to be in a scenario where you would really like to move your projects forward, but with the shelter in place or stay at home orders, if you're not able to get into the lab, we have our two arms, academic assay services and pharma assay services. I've shown academic assay services here, where you can send our samples to us and we'll perform the assay for you. This could include doing the proof of concept, limited resources, or if you're in a situation, as I mentioned, not being able to get into the lab because of the COVID-19 situation, we are here to do the assays for you. And obviously, this assays would have a quick turnaround. This can be a pilot project before you scale up, or it could be a regular project that you would basically like to get results for grant applications or paper submissions. And again, please contact technical support using the email address shown here for further details. Similarly, if you are from the biotech or the biopharma world, we have our pharma assay service team, which can also take in samples from you to perform the assays. And this would include pre-qualified tissues and sourcing, embedding and sectioning the samples, performing the staining, performing visual scoring or digital image analysis, have your images reviewed by pathologists if that's required, and finally have a report generated for your data. So again, I would like to reiterate the fact that if you are not able to get into the lab to move your RNAscope related projects or BaseScope related projects forward, we have the academic and pharma assay services team here on site, which is functional, and they can do these assays for you while you cannot get into the lab because of the COVID-19 situation. With that, I come towards the end of my webinar for today. Once again, I would like to thank you all for joining today's webinar, and I hope you found it useful. Shown on this slide are multiple ways of contacting technical support. You can contact us via email at support.acd@bio-techne.com or via phone using the phone number shown here. We are available from 8 a.m. to 6 p.m. Pacific Standard Time. And obviously, you can visit this link to get support-related resources. And finally, I would like to let you all know that I still have my colleagues who are on the line who would be able to answer the questions that you have. And again, if you have any questions, please direct your questions to all panelists in the Q&A window. And again, I would like to thank you all for attending today's webinar. My colleagues, as I said, are on the line to answer the questions that you have, so feel free to send in your questions via the Q&A window.